All right, guys. This one should be quick. All right, today we're going to talk about the cell membrane and cellular transport. And I want you to look at this incredibly complex picture of the cell membrane. Oftentimes we just draw it as a line, but I want you to see how complex this thing is. We talked about macromolecules, and you could see one of every macromolecule that we talked about. Here's a carbohydrate chain. Here's a protein, right? Here are your phospholipids. Well, I can't see DNA. But the cell membrane is incredibly complex. Okay. All right. Before we get into it, let's talk about homeostasis. You should remember this as one of the characteristics of life, right? Homeostasis is the need of an organism to maintain and regulate a constant or stable environment, internal environment. Much of homeostasis is maintained by the cell membrane because the cell membrane controls what enters and leaves the cell. Things that we need to control are enzymes, pH, concentration of other materials, and nutrients, right? If you think back to the conditions which are necessary for an enzyme, you will remember that an enzyme is very specific. It works best at a particular pH, at a particular temperature, right? And so these things have to be regulated within a cell in order for everything to happen as it should. All right. So the cell membrane is said to be semi-permeable or selectively permeable, means that some substances can move across freely while others have to go through a gate, right? The transport of materials can be classified as either passive transport or active transport. So passive transport does not require any extra energy by the cell because the molecules are moving from a high concentration to a low concentration. So think of a really crowded room and then the door gets open. Everybody's getting out of there. Okay. Active transport does require extra energy in the form of ATP to be spent to bring materials into the cell or to get them out. Active transport usually moves material from low concentration to high concentration. So it usually moves material against their concentration gradient. Some related vocabulary that you guys will find in this module. Solute is the thing that gets dissolved. So think of making lemonade. A solute would be the sugar or the lemonade powder. Solvent would be the thing that's doing the dissolving, the water. Solution is the uniform mixture of the two or more substances. So the actual lemonade that you made is a solution. Concentration is the amount of solute dissolved in a solvent. So Chick-fil-A's lemonade, very concentrated. McDonald's lemonade, not so concentrated. I'm kidding. So a high concentration is something, if it's lemonade, that would be really, really sweet. A low concentration is something that's not as sweet. Hypertonic solution is a solution in which the water concentration is below what is found in the cell's cytoplasm. All right, so water will tend to move out of the cell, all right, down its concentration gradient, and that's going to cause a cell to shrivel. A hypotonic is when the water concentration is above what is found in the cell cytoplasm. So there is more water outside the cell than is inside. And so again, water is going to move into the cell because it's going to move down its concentration gradient. So that's going to cause a cell to grow. So if a cell is in a hypotonic solution, it will grow, right? If a cell is in a hypertonic situation, it will shrivel because there is more water inside the cell than outside, so water is going to move out. All right. Isotonic is your best friend. Isotonic solution, the water is identical. The concentration of the water is identical to what's outside the cell. So animal cells typically love to be in an isotonic situation. You might want to write this down. Um, while plant cells prefer to be in a hypotonic situation because they have a cell wall and their cell wall is able to prevent them from bursting. Passive transport, there are different types of different transport. First that we're going to look at is diffusion. Diffusion is the spreading out of molecules across the membrane until they are equally concentrated on both sides. Right? So in this first frame here, you see that the molecules are really concentrated here and there is nothing happening on the other side. So what happens is over time, the molecules cross the membrane and eventually the goal of diffusion is for everything to be the same. So if Alyssa, one of the Alyssas, is in a corner of my classroom and she sprays her wonderful cologne, eventually the people at the next end of the classroom is going to smell it. So the people where she sprayed it is going to smell it really strong over here, but then eventually the whole class will smell it and it probably won't be as bad as the people who were in the corner where she sprayed it. And facilitated diffusion is where you have a transport protein that acts as a channel. So you have a protein channel to help facilitate the diffusion of the molecules. Normally, these molecules wouldn't be able to pass to the cell because either they're too big or they're too polar. If you guys take it back to when we discussed polarity is having different and unequal distribution of charges. Molecules, again, move down the concentration gradient from high to low, right? And things that use facilitated diffusion are glucose and sodium. So glucose is too big and sodium has a charge. So that's why these things need a special gate to go through. Osmosis. Osmosis is diffusion of water across the membrane. So water will move down its concentration gradient from high to low. So again, osmosis is pretty much the same thing as diffusion, but when you're talking about osmosis, you are looking at the actual water molecules. You're not looking at the solute. Okay? So in this picture, you can see here that so this is a selectively permeable membrane, right? And you can see on this side it's really concentrated with solute, but on the other side of it, you see that there's more water, right? So technically the water is more concentrated. So water itself is gonna move from the area of high concentration of water molecules to an area of low concentration of water molecules. So eventually you'll look like this and you'll see that what has moved is not the solute, but the solvent. All right, so the water is going to move so that this concentration can be equal on both sides. So taking what we've talked about with, hypo, hyper, and isotonic, I want you guys to really think about what is happening in this picture and why is the salt a weapon to a slug? All right, so think about it. Ask me about it in class in case I forget to discuss it, and we'll talk about what will happen to the slug and why in terms of concentrations and solutions and all that. Active transport. So as I said earlier, not everything is going to move freely into the cell, right? Because some things are too big or too charged or whatever their reason. Some molecules have to be pushed against their concentration gradient, and this is going to require energy. So from an area of low concentration to an area of high concentration, right? remember that we said it is natural and it is normal for you to want to move from a crowded room to spread out, right? It is not natural or normal for you to want to go where it's not crowded, so you're forcing your way into a crowded room, so to speak, right? Um, so other substances are too big. This is accomplished by either endocytosis or exocytosis. So those are two of our main active transport processes. Molecular pumps is another way that we accomplish active transport. This is when you have like a protein pump or a protein channel. So remember, we have protein channels that do facilitate the diffusion. So what you're going to look for is it using energy? And in this picture, you could see that ATP is needed for this to happen. So what this does, it allows key molecules to concentrate within the cell. So in this scenario, the natural movement of hydrogen ions would be to move from this part where I'm pointing to that part, where there's a lot of hydrogen ions to where there's not a lot. But in this case, we need all the hydrogen ions, and so this is when it has to be pumped using a protein pump. All right. Other things that we transport this way are like calcium or potassium, chlorine, sodium, and notice that all those are charged particles. Endocytosis, the cell uses energy again to import large amount of materials into the cell using a vesicle, right? There are two types of endocytosis. Could be phagocytosis, and that's when the cell is taken in solid particles, like in this picture here, and like what the amoeba does, or penocytosis, which is when it takes in fluid from outside the cell. So in both of these cases, the cell is importing material to the inside of it, all right, and forming a vesicle. So from outside to inside. Endo means into. The next one is exocytosis. Word part exo means outer, right? Exosphere, right? Means outer. The cell uses energy to export a large amount of materials out of the cell using a vesicle. So both of these use vesicles. One just takes things in, the other takes things out. And here's a picture of what that might look like. So here's the vesicle. It fuses with the cell membrane and it kind of gets the thing out. The best example of a cell that uses exocytosis is your nerve cell. So here's one. Here's the vesicle. It fuses with the membrane and it gets the thing out of the cell. Of course, on the other side of that, you'll have endocytosis happening to get it into another cell to transfer the signal to your brain. All right, so that is it for cellular transport, guys. Make sure you write all your notes, make sure you draw the pictures for clarification, and make sure you come to class with any questions that you might have.